Test. Okay. Very good. All right, folks, we finished going over the drugs that are used to treat diabetes. We really have only two main sets of drugs to cover. And that would include, oh, good gravy. Drugs that are used to treat cancer. And then we've got kind of some cleanup things to do, bladder drugs, some hormone drugs, things like thyroid drugs and what have you, which those won't take long at all. And then if we have time, I'd like to talk a bit about drugs for the eyes and the ears, because those are drugs you'll administer quite a bit probably in your careers as nurses. But for today, a group of drugs that ends up being encountered on a regular basis on the NCLEX are the anti-cancer drugs. And this is not a group of drugs for the faint of heart. Not by any stretch of the imagination. No way, no how, no nothing. So without further ado, let's just charge right in. So the anti-cancer drugs are classified according to their mechanism of action. Alkylating agents form adducts with DNA. They damage DNA, they prevent DNA replication, they make the DNA unrepairable, and because the tumor cell, which is dividing very quickly and is therefore very susceptible to inhibition of DNA replication, the cell will eventually give up the ghost and kick the bucket because it cannot divide properly, can't repair its DNA, and it can't traverse through the cell cycle. The anti-metabolites are drugs that prevent the synthesis of nucleotides. Since you can't properly synthesize nucleotides, you once again are left without the raw materials to be able to replicate your DNA or make RNA for that matter either. So the cell essentially gets starved of nucleotides and it cannot repair its DNA, replicate its DNA, express genes or things like that. The alkylating agents, remember, will include drugs that have platinum inside them. So cisplatin was actually developed up the road at Michigan State, and it revolutionized the treatment of solid tumors, hands down. So it and its derivatives are types of alkylating agents. They're a bit off the beaten path because they don't really have alkyl groups, but they modify DNA in ways that are very similar to what you see with alkylating agents. Then you have the so-called anti-tumor antibiotics. Bit of a strange name, I suppose, for drugs, but these drugs are going to form complexes with DNA. Very often they'll form complexes with iron. So they'll bind iron and DNA, and then they use the electrons from the iron to basically shred the DNA to pieces. Pretty effective. Hard to grow, hard to be a cancer cell when your DNA has basically had a cheese grater taken to it. So that's essentially how those drugs work. You have other groups of drugs that are not listed on here, groups of hormones. Some cancer cells are very susceptible to different types of hormone mimics, hormone antagonists, things like that. You have another group of drugs that are monoclonal antibodies. They bind to specific tumor antigens and they activate the immune system against the tumors. Other types of monoclonal antibodies are part of this smaller group of medicines that are called immunotherapies. Tumor cells, particularly solid tumor cells, make cell surface molecules that tell the immune system, leave me alone. So when you're at the hotel and you have a little sign you put on your door that says, do not disturb, that's kind of what these molecules are. They're a do not disturb sign for them and the immune system. So T cytotoxic cells or natural killer cells come up to the tumor cell and they see the do not disturb. And these molecules basically tell the immune cells, shh. And that's what they do. They go off and they don't bother the tumor cell. Well, essentially what these immunotherapy cells do is they take the do not disturb sign and they rip it to shreds. And then they take a sign and they hang it on the tumor cell that says, kick me. And then the immune cells come in and do just that. And they kick the crap out of the tumor, essentially. And if awakened against a tumor cell, T cytotoxic cells and natural killer cells can do a monstrously good job of destroying them. And that's what immunotherapy does. They are a subset of the monoclonal antibodies. Then you have groups of small molecules that are made against certain types of receptor tyrosine kinases that go awry and get illicitly activated inside cancer cells. You'll notice that many of these end in NIB, NIB. They are inhibitors. They're inhibitors of these tyrosine kinases. Many of these are made so that if the cell becomes resistant to one type, you can transition to another type. So these tend to be more specific for these rogue types of growth factor receptors that go rogue, so to speak. You then have other groups of anti-cancer drugs that inhibit microtubules. Microtubules are very important for making spindles. And there are several groups of drugs, well, two main groups of drugs that inhibit these microtubules. And then at the bottom of your handout on the last page, there's kind of this grab bag of stuff. And these don't rightly fit anywhere. So they're kind of in this miscellaneous group. A couple of these are going to inhibit topoisomerase. Some will inhibit ribonucleotide reductase. And then others work in various other ways as well. So that's kind of the Google satellite view of the anti-cancer drugs. Cancer, of course, is a very active field of research. And if we look, for example, just in 2014, you can see that there are several drugs that the FDA approved in 2014. All of these are just for cancers. And notice that they're for specific types of cancers. My dad and I used to argue, and he used to say, well, they've been working on cancer for all these years. Why haven't they cured it? And my rebuff would always be the same. Which cancer, Dad? Which one? Cancer is a grab bag for lots and lots and lots of different diseases. I don't think I ever got Dad to understand that. But till the day he died, that was always his line. They've been working on cancer for years. Nixon declared war on cancer. Why haven't they fixed cancer yet? Well, there's been quite a bit of progress made on blood type cancers, leukemias, lymphomas, particularly in children. Solid tumors, they're a tough nut to crack. There is some progress that's been made on solid tumors, but it's been much more slow going. I don't know if I told any of this. My PhD work was on cancer biology. So this is a field I know something about. This is a picture taken from a review that was published in the journal Cell. Robert Weinberg is one of the premier cancer biologist of his day. He's retired now. So was Douglas Hanahan. Douglas Hanahan passed away, unfortunately. Hanahan pioneered the use of transgenic animals to study cancers, animals that were genetically engineered to develop certain tumors, and then those could be used as models to test certain types of therapeutics and what have you. Weinberg was one of the pioneers of tumor suppressor genes and oncogene research and what have you. This particular diagram shows you essentially some of the different types of anti-cancer drugs that have been developed essentially and what they attack. So let's rehearse some of the features of cancer cells, shall we? This might give you an idea of why some of these agents are effective against cancer cells. So if you put regular cells in culture, those cells will spread out and they'll grow until they're all in contact with each other and they fill the plate and then they'll stop growing. That is called contact-dependent inhibition of growth. Virtually all normal cells show this property. They don't pile up on each other. They get those contacts, they know they're in contact with each other, and then they say, I'm done. They're done growing. Cancer cells, if you put them in the same dish, they'll extend, they'll fill it, and they'll fill the dish, and then they'll start piling up one on top of the other. They have lost cell contact dependent inhibition. That's one of the first features of cancer cells. Number two, there's a very old assay for cancer cells, still used today. If you take normal cells and try to grow them in soft agar, they can't grow. They need something to attach to. They need to be rooted in something. Cancer cells don't care about being rooted, they just grow. So they grow in soft agar. They don't need to be rooted, don't need to be adhered to some sort of substratum, don't need to be in contact with another cell. They don't care, they don't give a rip, they're long rangers, they just grow. There's a sermon illustration in there somewhere, but I'll save that. 3, cancer cells secrete cocktails of proteases. Now, some normal cells will secrete proteases, but cancer cells are protease secretors par excellence. This is how they break down barriers. They break down basement membranes. That's how they invade other tissues. They secrete lots and lots and lots and lots of proteases, far more than any normal cell, non-cancerous cell. 4, cancer cells make angiogenic factors. Who can tell me what an angiogenic factor is? Yeah. These are factors that cause the blood system to remodel itself and bring a blood vessel to feed this growing tumor. So they make angiogenic factors. This causes remodeling of the blood vessel system. That's why surgical extirpation of cancer requires a lot of skill. You have to tie off these newfangled blood vessels. Sometimes many surgeons will cauterize them so that they're essentially dead ends, and the body will tend to resorb them. 5, cancer cells have a lot of things wrong with them, but they have turned off the systems that induce programmed cell death. Many cells, if they get damaged beyond a certain point, they can't repair that damage, they will, excuse the expression, off themselves. They commit huddy-cuddy, essentially. They commit seppuku. Cancer cells don't do that. That system has been shut off. And then 6, the main controls on cell division and the main checkpoints that regulate cell division have been hamstrung. So they just grow and grow and grow and grow and grow. Usual checkpoints, oh, Are all your chromosomes attached to proper spindles? Is your genome without damage? Have we counted the chromosomes? Do we have enough chromosomes? Can we replicate our DNA properly? All those checkpoints, hamstrung. Cancer cells just divide. They've got issues, they don't care. So Sunday, I was asked to officiate a wedding. And we were getting ready to start, and this homeless lady came right into the ballroom, and she was having a very spirited conversation with someone I couldn't see, at least. So her interlocutor was either invisible or just didn't exist. And she was quite upset at her interlocutor, and she kept going around and she was swearing. And I said, okay, if I get to the vows and this lady is making a scene, I'm going to ask an usher to escort her out. This is her wedding. You want that at your wedding? You want that on the videographer to videotape that? Have somebody swearing in the background? That's what I said. So I told the usher, I said, watch her. If she doesn't leave, politely ask her to leave. She won't leave, escort her. And if she raises a stink and gets violent, call the cops. So she is like a cancer cell. She doesn't care if she's causing trouble. There are protocols that you would observe. So Jenna, if you were lost, you would probably go up there and you saw a wedding kind of thing. You'd probably go.
I wouldn't go in.
Well, you'd say, or something like that. You wouldn't have a conversation how mad you were about how lost you were, and it's everyone else's fault, and start swearing and stuff like that. Please don't.
I would walk in.
You would walk in.
I would.
Okay. Or maybe if you have to rush usher or something like that. This usher's just standing there and you need something to do. So, well, because you understand protocol. She didn't understand protocol. That's what cancer cells are. The protocol for cell division, they don't care. They don't care. They're like a homeless person, essentially. So, given that to be the case, these are some of the things that have been designed, and some of these drugs are experimental. Some of these drugs are experimental at the time and are now part of the anti-cancer armamentarium. We're going to go over the more basic drugs, the more specialty drugs. If you guys get into that field, you'll study it then. So we'll just do the more mainline ones. But I want you to see that while cancer gives you a knight in shining armor, there are kinks in that armor. There are seams in that armor where you can get a very thin sword and inflict damage and maybe even kill it. So that's essentially what we're going to cover. We'll come back to this. Cell cycle. It's been a while since you've heard about the cell cycle probably, but let's talk about the cell cycle anyway. So the cell cycle is broken into two main parts. It's broken into an M phase you don't work. It's broken into an M phase, and that M phase is mitosis. And then everything else besides M phase is interphase. Interphase is then broken into three other phases: a G1 phase, an S phase, and a G2 phase. S stands for synthesis. This is the phase during which you replicate your DNA. It's not like bacteria where you replicate your DNA all the way through the cell cycle. You only replicate DNA during the S phase. G1 and G2 are either gap phases or growth phases, depending on whether you call them growth or gap. And this one is a phase where the cell recovers from mitosis and prepares to replicate DNA. There's a checkpoint right here. That checkpoint essentially will determine if the cell has the energetic resources to replicate DNA. Replicating DNA is very energetically expensive. If it does not have the resources, it will not put those resources into the S phase. Instead, it will go into a G sub 0 phase where it takes a nap. Comes out of S phase, then G2 is preparing for mitosis. There's a checkpoint right here in the middle of G2. It essentially asks, are all the chromosomes properly replicated? If they're not, let's pause a bit. Let's get things repaired. Let's get them fixed, and then let's move on. And then in the middle of M phase, before the cell goes from metaphase to anaphase, it says, is every chromosome properly linked to a set of spindles? That's the M phase, metaphase checkpoint. So there's three. One here, one here, one here. Some drugs can kill cancer cells in any phase of the cell cycle. Some are cell cycle specific. And you see this here. These drugs here are virtually all, except for hydroxyurea, which is missing an A. These drugs are virtually all antimetabolites. They work during S phase. These drugs here, most of these drugs inhibit topoisomerase. They're topoisomerase inhibitors. These drugs here are going to disrupt your microtubules. They act during M phase. Then you have others. These are all alkylating agents. They don't care where the cell is in the cell cycle. They can kill it anywhere, any place, anytime. So this is meant to give you an idea of how certain anti-cancer drugs work in terms of their specificity. With regard to the cell cycle, we'll talk more about that as we talk about the individual drugs. You guys have probably experienced this with people in your family who've had cancer. You get an additional treatment, you get an initial treatment. After that initial treatment, the tumor gets knocked down, and then some of the cells come back. They form maybe secondary tumors, and those cells are resistant to that initial set of chemotherapeutic agents. Cancer cells are very good at adapting. How do they adapt to anti-cancer drugs? There's several ways. First of all, they tighten their membrane. They don't let the anti-cancer drug in. Secondly, they use the P glycoprotein. They activate that gene, and they take the cancer drug and they pump it right out of the cell. So the cancer drug can't get a high enough concentration to do anything inside the cell. Three, the drug somehow does not get metabolized into its active form. If the cell needs to convert it into its active form, the cell just doesn't do it. Some cancer cells actually make enzymes that will inactivate anti-tumor drugs. A somewhat common strategy you see in various types of tumors is that the target is encoded by a certain gene, and the cancer cell takes that gene and amplifies it. So now when the cancer drug enters this cancer cell, the target is just too numerous for the drugs to work. That's called gene amplification. Gene amplification. 6, the target gets changed by mutation so that the anti-cancer drug no longer recognizes it. 7, there's one biochemical pathway that got shut down. There's an alternative pathway now that is turned on. That becomes a major pathway. So the anti-cancer drug now no longer works. Since it worked on a main pathway, the alternative pathway now is taken over. 8, there's a decrease in topoisomerase 2 and various DNA breaks. 9, DNA damage repair enzymes are upregulated. So your damage repair just gets repaired a whole lot faster. And this table comes from a review, and I shamelessly stole that table. This just essentially is used to show you the different
types of mechanisms that are used. Very good. Let's get to the agents themselves, shall we? So the alkylating agents cross-link DNA. They usually have two sites on the molecule that are able to bind DNA. One of the common targets is the nucleotide-based guanine. By cross-linking DNA, you prevent DNA from being separated for replication. You also prevent it from being separated for RNA transcription. Loss of DNA replication, loss of transcription leads to cell death. That's how alkylating agents work. Alkylating agents are cell cycle non-specific agents. Cell cycle non-specific agents. So first we have the nitrogen mustards. The nitrogen mustards. The nitrogen mustards include cyclophosphamide, I-phosphamide, meclorethamide. There's some other there, but those are the three I want to talk the most about. Nitrogen mustards are related to the poisonous gas phosgene. Any World War I history buffs out there? Phosgene was the poisonous gas used by the Germans during World War I. Nitrogen mustards are derivatives of phosgene. These are given typically intravenously. These agents are usually prodrugs, so they're administered in an inactive form. So this essentially gives you the mechanism of action we just talked about. The upshot is that you cross-link DNA. That's a piece I want you to see. So cyclophosphamide. There it is. There. Cyclophosphamide and I-phosphamide are used against leukemias. They're also used against other solid tumors like ovarian and breast cancer. Low-dose cyclophosphamide is also used to treat certain types of autoimmune diseases. It's used for progressive or refractory cases of systemic lupus erythematosus. It's also used for multiple sclerosis and several others as well. All right, in terms of toxicity, biggest toxicity with alkylating agents is bone marrow depression. and they can cause aplastic anemia. That is the biggest caveat with these drugs. Other common toxicities with these medications include hair loss, alopecia, gastrointestinal disturbances, and they can depress gametogenesis, which can cause infertility. Nitrogen mustard specifically will cause the syndrome of inappropriate antidiuretic hormone secretion, which is SIADH. So too much antidiuretic hormone will tend to cause you to retain liquids and raise your blood pressure. You treat it with drugs known as Vaptans, VAPTANS, Vaptans. Cyclophosphamide can also cause hemorrhagic cystitis. That's pretty common. If you keep using it, it can cause bladder cancer. Fortunately, there is a compound called sodium 2-mercaptoethane sulfonate, which is mercifully abbreviated MESNA. When administered with cyclophosphamide, it will tend to spare the bladder. All right, let's move now to, okay, yeah. Oh, here's the structure of MESNA. The trade name of MESNA is Mesex. This is given either by mouth or by injection into a vein. Allergies to MESNA do occur, so watch for that. Initial reports say it's safe during pregnancy, but that was a small study. I'm not sure I trust that. All right, so ifosfamide also causes hemorrhagic cystitis. It causes less hair loss and it's less hard, it's not quite as hard on the GI tract as cyclophosphamide. And it gets used for testicular tumors and adult sarcomas. There it is, there it is. Okay, moving on to busulfan. Busulfan, unlike cyclophosphamide is a prodrug, has to be activated. Busulfan does not need any activation. This particular drug spares other organs quite nicely, but it's really hard on the bone marrow. So it's one of the most potent agents against leukemia. It's particularly effective against chronic myelogenous leukemia. Pleasure. So you'll see suppression, you'll see decreased numbers of platelets. That means bleeding is going to be a risk. You'll see decreased red blood cell counts, so anemia, and all of its caveats, shortness of breath and things like that, pallor, and decreased numbers of white blood cells, so increased risk of infection. Let's talk about the nitrosoureas. These drugs are highly lipid soluble, so they cross the blood-brain barrier. Therefore, they are used to treat glioblastoma multiforme. So these two drugs include this drug shown here, carmustine. The other is lomustine, carmustine and lomustine. I swear as it gets later during the day, my handwriting deteriorates from bad to really bad. They cross the blood-brain barrier, but that means they're neurotoxic. So they can cause convulsions, seizures, ataxia, poor coordination, and things like that. That is all I'm going to say about the alkylating agents. I'd like to talk a bit about the platinum-containing agents, which work a lot like alkylating agents. No course on pharmacology would be complete without talking about cisplatin. Again, cisplatin, like the other alkylating agents, will kill cancer cells regardless of where they are in the cell cycle. There are three main platinum-containing agents. There is cisplatin. There's carboplatin. And then oxaliplatin. Emily, is that big enough? You can see that? Erica, you can see that? Haley's, okay. Not everyone's as blind as I am. These are also administered intravenously. As we said before, cisplatin revolutionized the treatment of solid tumors. It also targets guanine, and the two chlorines there, they get displaced, and these will also cross-link strands of DNA. Cisplatin, so enter cells, you get the highly reactive platinum complexes. Platinum withdraws electrons from chlorine, which is quite a trick when you consider chlorine because it's a halide, which has a pretty high affinity for, well, excuse me, it gives electrons actually to chlorine. Chlorine withdraws electrons from platinum. Platinum is rich in electrons, it's a metal. And then the chlorines essentially will leave, and platinum then reacts with the number 7 nitrogen of guanine, cross-linking it. You get interstrand and intrastrand cross-links of DNA that look like this. And that sort of thing doesn't lend itself to either DNA replication or transcription. Platinum also will cross-link proteins to DNA. Cisplatin and carboplatin tend to be used for a variety of solid tumors, including testicular tumors, bladder tumors, ovarian and cervical tumors, and lung carcinomas. So bladder, testicular, ovarian, cervical, and lung carcinomas. Oxaliplatin has different indications than either carboplatin or cisplatin. Oxaliplatin is used to treat advanced colon cancer, cancer of the biliary tree and liver, pancreatic cancer, and ovarian carcinomas. It also gets used to treat lymphomas. Cisplatin and carboplatin are also used to treat osteosarcoma in teens and children. There's carboplatin there. That's a structure of carboplatin. The platinum-containing drugs in general cause relatively severe GI disturbances, very severe nausea and vomiting. Ondansetron is a very effective drug against platinum drug induced nausea and vomiting. Have that on hand. Zofran. Have that on hand. It also can cause serious nephrotoxicity. You prevent nephrotoxicity with adequate hydration. That's going to be hard if your patient's up chucking front, left, and center. They're not going to want to drink. They get dehydrated because of all the up chucking. You're going to have to hydrate them and through another route, if you know what I mean. You can induce diuresis with mannitol. Mannitol will get more fluid flowing through the kidney. That prevents the drug from building up in the kidney and causing damage. You can also use normal saline. There is also a compound you can give that will scavenge reactive oxygen species in the kidney and diminish kidney damage. It is called amifostine. Amifostine. Another common side effect of the platinum-containing drugs is peripheral neuropathy, usually in the fingers and also in the toes. Ototoxicity is another feature. When you have patients on platinum-containing drugs, you need to be monitoring their hearing routinely. If the patient gets a ringing in their ears, that might be the first sign of hearing loss. So of the platinum-containing drugs, cisplatin is the hardest on the kidney and also causes the most neurological side effects. Carboplatin is not as hard on the kidney or nervous system, but it's harder on the bone marrow. Oxaliplatin causes less ototoxicity, less nephrotoxicity, less bone marrow suppression than either cisplatin or carboplatin. Let's go ahead and take our break here, and then we will come back and I will talk about the topoisomerase inhibitors. Are you going to be in the pinning ceremony? No, that's going to be at White Auditorium before this the ceremony here.
Yeah, it's before graduation, so it's like super early.
Super early as an 8 or
I think it's like 8 or 9.
okay. I really want to go. Mindy asked me if I could be there. I said, see, I'm a marshal for that, so I have to show up early.
Yeah, we have a breakfast at 8 and then the pinnings at 9. Yeah. And that's in the White Auditorium for both the
Darn it.
breakfast and the pinning. And then the graduation is obviously at 10.
Is it 10? Yeah, and I'm supposed to be there at like 9.
No. We can go to the breakfast at 8 a.m. That's okay. So that's what I did. I had a couple like this. Yeah. It's like, well, you said I leave Washington. Yeah. Yeah. Okay.
I have this.
Hmm.
And is it okay if I take the chapter 12 quiz now, or should I wait? Not chapter 12, but quiz number 12.
I suppose you could take it now. McKenna, it's on the printer. The office is probably locked. Get it.
Usually, 40 at least. 2 days. Once at least. I took one last. Wow. Why did you turn it off?
Do that to yourself, please.
Why would you? Why would you? Normally they like.
Can we discuss this with the other thing?
Yeah. Send me an email to remind me.
Why are you lying? Yeah.
Real. And that's third. And then, Emily's-
Oh, yes.
No.
No,
Greenville. Greenville. So they get to go to their national. She was very excited. They had the last match. I remember one person was sorry. And she's been playing really well. I had nice to see her. So she won her both her doubles match and her singles. Yeah. We'll take to their club. All right, let's get back in the saddle. So as a quick review of DNA replication, you have the DNA helicase that unwinds the two strands of DNA. You then have one strand where DNA synthesis is continuous. So you might have, say, a primer like this, and then you get continuous synthesis. So that's a leading strand. Synthesis on this strand will go as fast as the helicase can unwind the DNA. Then you have a lagging strand where synthesis is going to be discontinuous. And so on and so forth. Then ahead of the helicase, you have topoisomerases that are taking the DNA and getting rid of three-dimensional structures that can add more tension to the molecule that prevents the helicase from being able to pull it apart. Without topoisomerase, the helicase will slow down. It'll eventually get to the point where it can no longer unwind the DNA. The DNA will have these superstructures, knots even, that the helicase cannot separate. DNA replication will stop, and because the DNA can't be replicated, the cells can no longer divide. There are two types of topoisomerases in eukaryotic cells, topoisomerase 1 and 2. Topoisomerase 1 makes one of the two strands. It cuts one of the two strands. So it cuts a single strand, lets the DNA essentially unwind, and then reseals it. Topoisomerase 2 will make double-stranded cuts in the DNA. So there are groups of anti-cancer cells that inhibit topoisomerase. The first group are the so-called camptothecins. And I think I have those around here somewhere. Oh, no, not there. There we go. The camptothecins come from the stems of the so-called Camptotheca tree. This tree also is called the tree of life, interestingly enough. So the camptothecins include
irinotecan and topotecan. These inhibit topoisomerase 1. So the camptothecins. They inhibit topo 1. Irinotecan is a prodrug. It has to be metabolized into its active form in the liver before it can work. Irinotecan is going to be excreted through the bile and through fecal material. So you don't have to adjust it for patients with compromised renal function. Topotecan is excreted by the kidney. Therefore, its dose will need to be adjusted according to the renal function of the individual. Irinotecan is used to treat metastatic colon cancer. Topotecan is used to treat advanced ovarian cancer. And it's also used for small cell lung cancer, small cell lung cancer. Most common side effects of these drugs are myelosuppression, depression of the bone marrow, and then gastrointestinal issues like diarrhea, which is more prevalent with irinotecan than it is with topotecan. In general, these particular drugs are better tolerated than a lot of other anti-cancer drugs. Next, you have medicines that inhibit topoisomerase 2. Topoisomerase 2. These drugs are called the podophylotoxins. Podophylotoxins. So this includes these drugs that are listed here: etoposide and teniposide. Notice these end in poside, whereas the camptothecins end in tecan. Tecan. These drugs are both eliminated through the kidney. So you're going to have to reduce their dosage. The topoisomerase inhibitors in general are going to be cell cycle specific. That shouldn't surprise you. What part of the cell cycle do you think they'll be active? Very good, Natalie. Synthesis. Well done. S phase. S phase and G2 phase. Because to repair DNA, you also have to replicate it, very often to repair it. So S phase and G2. Common toxicities of these drugs will include, again, myelosuppression, gastrointestinal disturbances, alopecia. These drugs are used in testicular and small cell lung cancer, and they're also used in leukemia. Jumping around a bit, but hey, that's all right. Okay, let's take a look at drugs that inhibit microtubules. Microtubules. Microtubules are part of the cytoskeleton. They consist of a polymer of a dimer called alpha and beta tubulin. Alpha and beta tubulin. These two proteins make a dimer. They assemble together to make a hollow, stiff rod. That hollow, stiff rod is called a microtubule. It forms a dynamic network throughout the cell, but microtubules are really, really important for cell division. They form the spindle that will pull the chromosomes apart. Because these drugs interrupt the spindle, they are going to be cell cycle specific. They will work during M phase, during mitosis. There are two groups of antimicrotubule drugs. There are the vinca alkaloids and then the taxanes. The vinca alkaloids and then the taxanes. This is the vinca plant from which they were extracted. This is also known as the Madagascarian periwinkle. So these are actually found in the rainforest of Madagascar, and these were discovered actually through conservation work that was being done in Madagascar. And when these were discovered, it was, many conservationists said, well, see, we need to preserve the rainforest because there are compounds in these plants and things like that that might end up being economically useful. If we destroy the ecosystem, we won't know about. There are two main vinca alkaloids, vincristin and vinblastin. Vincristin and vinblastin. These bind to beta tubulin and they block polymerization of the filaments. So they prevent the microtubule filaments from forming. The cell cannot form stable microtubule spindles. They will cease at metaphase. They'll pile up at metaphase and then they won't divide and they'll eventually die. Vinca alkaloids are used for solid tumors like breast cancer, pediatric tumors like rhabdomyosarcoma, that's a muscle tumor, nephroblastoma, that's a kidney tumor, and of course, leukemias. Both vincristin and vinblastin are also used for Hodgkin's and non-Hodgkin's lymphoma in adults. All right, so these particular drugs disrupt cell division in hair follicles, so they cause alopecia. They also cause gastrointestinal symptoms. They are associated with bone marrow suppression, but vinblastin definitely causes more bone marrow suppression than does vincristin, but vincristin is more neurotoxic. The neurotoxicity is very much of a glove and sock sort of neuropathy. Vinblastin can also cause things like orthostatic hypotension, urinary retention, and also constipation. I'm not going to talk about that one. Next, we have the taxanes. Taxanes. These are alkaloid derivatives of compounds found in the bark of the yew tree. So the Pacific yew and the European yew tree. You can see the Pacific yew tree over here. If you go to the West Coast, this is a pretty common tree on the coast. If you drive up Highway 1, it'll take you through, actually it'll take you all the way up almost to Canada. And if you get out of your car and look around on the coastal forests that you'll drive through, you will see yew trees. They're present. You'll see more redwoods and things like that, but you'll also see some yew trees. There'll be smaller trees usually. And then there's a European yew tree that has these fruits that are very different from the Pacific yew tree. These work in a very different way than the vinca alkaloids. They will enhance tubulin polymerization, and then they cement the microtubules together so they can't disassemble. Chromosome separation requires that the kinetochore disassemble the microtubule. That's what causes it to move away from each other. These drugs, lock the microtubules in their stable form. So they are also cell cycle specific. They work during the M phase. These are used for breast, ovarian, lung, gastroesophageal, prostate, bladder, and head and neck cancers. You want me to repeat that? Breast, ovarian, lung, gastroesophageal, prostate, bladder, and head neck tumors. Their main side effects include, you guessed it, alopecia, myelosuppression, and neurotoxicity manifesting as a glove and sock neuropathy. Let me just say here as a nursing, something for nursing concern, when someone is experiencing myelosuppression, are you going to give them other anticoagulants? No, Natalie's looking at me as if to say, that's a really dumb question. But no, you wouldn't, would you? Because their platelets are already low. You give them anticoagulants, what are you going to do to their ability to clot? And they're really going to have bleeding trouble. So you need to not use these with other anticoagulants. All right, let's talk about the ribonucleotide reductase inhibitors. Let me remind you of something.
So this is ribose. If you want to make a base, a nucleotide that's fit for DNA, you need to do something. That hydroxyl group, what do you need to do to it? This is good for RNA. To put it into DNA, you need to erase it and replace it with what? It's deoxyribonucleic acid. So we want to get rid of the deoxy. That work? Yeah. And all nucleotides are originally made as ribonucleotides. All of them. Then they are converted to the deoxy form by the enzyme ribonucleotide reductase. So ribonucleotide reductase is a very, very busy enzyme inside the cell. And as you might guess, if you want to inhibit cells that are growing very fast, this would be a prime target, yes? In fact, it is. If you inhibit it, you don't make any you don't make any deoxyribonucleotides. Don't make any deoxyribonucleotides, can't make DNA. Can't make DNA, you can't grow. You can't replicate your DNA, you die. And if you're a fast-growing cell, that's going to be a big seam in your armor through which you can drive a very sharp sword. Now, here's the other thing about ribonucleotide reductase. This enzyme has been worked on extensively. There are excellent crystal structures of the enzyme. The enzyme mechanism is known to the nth degree. It's extremely well understood enzyme. So making inhibitors for it has been something that many biochemists have worked hard at. So there are molecules that will inhibit this enzyme quite powerfully indeed. Let's see, am I going the wrong way here with this, or no, I am going the wrong way. I'm jumping around a little bit, I'm sorry. Yeah, here we go. Okay, hydroxyurea. We've seen hydroxyurea before. Where do we see hydroxyurea in another class? In genetics, actually. What's it used for? Sorry? No, not leukemia. It is used for leukemia, yeah, but it's, what other conditions is it used for? Sickle cell disease. Remember? Sickle cell disease. It induces the synthesis of the fetal hemoglobin genes. And the fetal hemoglobin beta chain doesn't have the sickling mutation. So the fetal hemoglobin will displace the beta globin with the sickling mutation in it. It is also used as an inhibitor of ribonucleotide reductase. So ribonucleotide reductase has an iron cofactor. Hydroxyurea binds to that iron cofactor and gums it up. So this is used for disorders that are characterized by proliferation in the bone marrow.
So it's used for chronic myelogenous leukemia. It's also used for something called polycythemia vera. Polycythemia vera. And it's also used for sickle cell disease. Since it works in the bone marrow, its main side effect is myelosuppression. This tends to present as a leukopenia. Red cells are not as heavily affected, but anemia is a potential side effect. Also, occasionally thrombocytopenia. Other side effects include gastrointestinal disturbances, pneumonitis, and mild skin reactions. Oh, I saw it over here. Where are you? Oh, you're not going to let me. Okay, well. The other drug that will inhibit ribonucleotide reductase is a drug called fludarabine. Fludarabine. Fludarabine is given either orally or intravenously. In plasma, it's given as fludarabine phosphate. Then in plasma, it gets converted to fludarabine. When it gets in the cell, it gets converted to fludarabine triphosphate. That's the active form. And it binds directly to and inhibits ribonucleotide reductase. It has an additional ability to get incorporated into DNA polymerase and inhibit it. Fludarabine is used for leukemias like chronic lymphocytic leukemia and other low-grade lymphomas. Side effects include things like myelosuppression, fever, gastrointestinal disturbances, and that's about it. Oh, I thought I saw this over here. Yeah, there it is. Gemcitabine. Gemcitabine is intravenous. This drug enters cells. It gets phosphorylated. It gets phosphorylated into a diphosphate form. After that, it binds to ribonucleotide reductase and inhibits it. The triphosphorylated form can also get incorporated into DNA polymerase and block DNA synthesis. Gemcitabine is used for pancreatic cancer, non-small cell lung cancer, ovarian cancer, bladder cancer, and non-Hodgkin's lymphoma. Its side effects include myelosuppression, pulmonary toxicity that usually presents as a pneumonitis, and a kind of flu-like symptom. Long-term use of gemcitabine can lead to liver toxicity. Our time is escaping. Let's start the we'll just start these. We won't get through them. We'll just be able to start them. So these particular drugs, they're going to mimic purines and pyrimidines. They're going to get incorporated into those pathways that are used to make purines and pyrimidines, and they're going to inhibit many of the enzymes involved in the synthesis of purines and pyrimidines. So they are going to essentially really gum up the synthesis of nucleotides. That's how antimetabolites are going to work. The synthesis of purine and pyrimidines uses two main pathways. There's a de novo pathway where you make things from scratch. Very energy intensive, requires lots of precursors, and then salvage pathways. Salvage pathways take already used nucleotides and recycles them. Some of these will inhibit both. Some of these will inhibit one over the other. Some of these will specifically inhibit the salvage pathway. So methotrexate is a good example of a drug that is an antimetabolite because it masquerades as folate. Since it masquerades as folate, it inhibits dihydrofolate reductase. Dihydrofolate reductase is required for not only the synthesis, but also the recycling of tetrahydrofolate. Tetrahydrofolate is essential for single carbon transfer reactions. Single carbon transfer reactions are essential for the synthesis of thymine. Without tetrahydrofolate, you are dead in the water. And so that's how methotrexate works. It hamstrings tetrahydrofolate and prevents you from making thymine. Because you can't make thymine, you can't make DNA. It also prevents you from making the amino acid methionine, amino acid serine, and then these two purines, adenine and guanine. We looked at methotrexate as a drug for treating certain autoimmune diseases. When it's used as an anti-cancer drug, it's used at a higher dose. Therefore, its side effects are much more acute. All right, let's stop right there. We'll come back Wednesday. We'll finish the antimetabolites. We'll then look at the anti-tumor antibiotics, the monoclonal antibodies. We'll have our last quiz of the semester, and there was much rejoicing. And I looked at the forecast. The weather still looks pretty crummy, but next week looks nice. So maybe Monday we'll go outside. Yes. Wednesday we have class and that's our final. Next Wednesday, that's our final. Yeah. That's never stopped us in the past. I'll be here holding class. Yes, I'm so mean.
It's gonna be on like phone. It's also on devices. It's just like
Why
around.
don't you
right now.
Zoom?
No, it's fine. I still want to be able to take my quiz. I don't want to
Okay,
because I want to make up close to finals or anything like that.
you're gonna be in painkillers?
Yeah. I might have my mom or something. I mean, 'cause I really don't have to make it up. But it depends how I feel. I'll do the day out.
Do you only come to class once a week for this class essentially?
For this one?
If you come to campus just for the art, or you cross the street for the NCLEX reviews,
Not just today. I like here.
the rest of the time you're at the sites.
At my yeah.
So coming in to take the quiz is a pain,
Yeah.
because you have to make
I'll
an extra
just-
trip.
Yeah. I'll figure it out. I might be off the pain medications by Wednesday. I have a pretty high pain.
Okay, if you Zooming, I have no problem Zooming if you want to Zoom.
Okay.
And if you want to take the test administered by email, I'm okay with that.
Okay.
I trust you.
Yeah, I tried to find my 6 and 9, and I don't know if my mom threw away because she was spring cleaning in my room.
Uh-huh.
So I still haven't been able to find it. And I
Okay.
like rummage through. I have like a bookcase where I keep all my papers, and
Right.
I could not find it.
Okay.
So
We'll figure it out. All right. Don't be a hero. Don't be a hero. Seriously.
yeah, I don't think it should be that bad.
The procedures, sometimes they hurt a lot more than you think they're going to.
Yeah.
And sometimes you're pleasantly surprised. That's the exception usually.
We'll hope for that.
Okay. Yeah.
I was just asking, I still don't have my second grade. Not second grade, but the other half of my math work classes.
You sent them to me, right?
Yep. I
Yeah.
emailed you the link of the paper that I wrote about one that I watched.
And you sent it this weekend?
No, I sent it.
You sent it a while ago.
Yeah, it was quite a while. I mean, I can resend it to you.
Yeah, resend it. Just resend it because at this stage,